7, 6, 5, 4, 3, 2, 1.
Welcome to SIYSS 2023. My name is Julia Tollin, and I will be the stage host today on this seminar. I have the incredible honor of guiding you through the presentations today. And I'm very passionate about this, having young, brilliant minds coming to Stockholm, sharing their research and science from this stage today. And to get to know a little bit more about this seminar, we will be joined on the stage by Elin Palm. Elin, welcome to the stage. You are the project manager, and we, of course, want to make sure that everybody knows who are actually these young scientists. Who are the participants coming up on stage today?
Today we have 18 participants presenting the research they have been conducting. They come from 14 different countries or have 14 different nationalities. And they have been conducting science in all related subjects, from chemistry to physics, mathematics, biology and bioengineering. So really inspiring youth scientists here.
And I've been chatting a little bit with them. They're really smart.
They are so incredibly smart, and it's so amazing to listen into the conversations we have with them throughout the whole week and the discussions they're having. Super inspiring youth.
Okay, so if you're sitting in the audience right now and you're interested in science or technology, mathematics, these subjects, and you would like to be someone in the future who would be standing on a stage like this, sharing your research, how would that journey look like?
First, though, I want to say, go for it. It's amazing to be here as a participant. There are a couple of ways to get to be here and present your research on stage. I would say for most of you in the audience, you are most likely high school students, meaning that you will be creating or conducting your high school diploma project. And if you do that really, really well, and you compete with your high school diploma project in Utställningen that Unga Forskare is organizing the exhibition, then you might win as one of the prizes, or you might become part of the Swedish national team in science. And then you get to compete with your high school diploma project in international competitions, like the European Youth Competitions for Scientists, or something like that, or the International Science and Engineering Fair. And if you're super lucky and perform well in these international competitions as well, you might be the lucky winner of coming here to SIYSS and presenting your research here. The other way to go is that if you, after your high school years, decide to attend one of our associated or partner universities or sending organizations, like Luleå University or Cambridge, they are also selecting a few students to be coming here presenting their research. But I would go for a high school diploma project and then participating in Utställningen.
Cool, that's something everybody can do, right?
Yeah, more or less. You need to have a high school diploma project that is related to science and be proper research. I know I did my high school diploma project in literature research, and that was apparently not good enough to be able to do that. So you need to have like original research going on. But if you do that, which I really hope you will, and that you feel inspired to do listening to these amazing young scientists, then you can definitely compete in Utställningen. And there is nothing like, try, at least go for it.
Okay, so you're very engaged in the Federation for Young Scientists, who are arranging this seminar day and the SIYSS week. Tell us about this organization.
The Swedish Federation of Young Scientists is the biggest youth organization here in Sweden, and we are passionate about youth in STEM, and we want STEM to be an obvious choice and a possible choice for all youth across the country to engage in. So we do this by having partner members that are the local organizations that are across the country. But we also organize certain bigger events like SIYSS, the one you're visiting today. But we also have Utställningen that I've been mentioning a couple of times. We have summer research schools that I think could be a really good opportunity if you want to become one of our future SIYSS participants. And other activities going on throughout the year. And I also noted that we are handing out these little brochures right outside here. So if you're curious about what we are doing and how you could participate in it and how you can get help potentially in creating your high school diploma project or potentially becoming one of the future SIYSS participants, take a small brochure on your way out and that could give you more info.
Perfect. Give it up for Elin Palm. I have a sidekick today who is doing some really good stuff, taking care of the questions in the Mentimeter software that's coming up. Give it up for Arman. Have you gotten a lot of questions throughout the day?
Yeah, so basically people ask questions via the Menti link, and I will look at them and pick out the ones that I think we'll have time to go through. It's been really fun. There's been some great questions so far. And I'm excited. Also, I just want to make a quick shout out to Raise for Excellence, which is a summer research camp that's also under Unga Forskare. I'm one of the organizers, so if any of you is a second year, please apply.
What was the name of the?
Raise for Excellence.
Raise for Excellence. Apply for that. Okay, cool, Arman. It's time to kick off with the first presentation. This project consisted of developing a mathematical model for the floating arm, a kind of catapult, and building a physical one to test the theory. Okay, so give it up from France, Clément Vovard.
Hello everyone, my name is Clément and I was sent here by SIYSS, the European Union Contest for Young Scientists. I'm really honored to be in front of you today to present my project, developing and testing a Lagrangian model of the floating arm trebuchet. Now, I know you're all probably asking yourselves, what does that mean? Lagrangian model, floating arm trebuchet. So let's actually start with the second one. This is a floating arm trebuchet. It's a sort of catapult, and I think it will be easier to understand if I just start with a demonstration. Okay, ready? Oh, wait. Yeah. There we go. Okay, so this wonderful machine was the focus of my project, and I wanted to understand its behavior both from a theoretical and experimental approach. So first, theoretically, I started by developing a mathematical model for it, which was broadly divided into two parts. The first part was modeling the trebuchet itself. This was done using something called Lagrangian mechanics. So Lagrangian mechanics, it's this alternative formulation of classical mechanics, which is more complicated than the usual Newtonian mechanics you might have heard of in high school. But it's a lot more powerful, so it's able to describe more complex systems like the floating arm trebuchet. I won't bore you with the details, but mathematically, Lagrangian mechanics relies on this equation, which is called the Lagrangian, which uses the kinetic and potential energies of the trebuchet to describe the system. Then, after a few pages of calculations, you eventually arrive at this, which is a system of 9 differential equations, which you can solve to model the trebuchet. Okay, so now that I have this, I can move on to the next part of my model, which was modeling the trajectory of the projectile itself. So intuitively, we all know how a ball moves through the air when you throw it. And here, I just have to replace this person with my trebuchet. Again, since this was, I took air resistance into account, this was determined by differential equations, but it was much simpler than the first part. Cool, so now that I have the two parts of the theoretical model, I want to combine them into a complete model. And for that, the link is the launch condition. When exactly does the projectile leave the trebuchet? This might seem easy, but it was actually one of the trickiest parts of the model to figure out. But eventually I discovered that there was this equation that governed the launch. Okay, now I have everything, so I can just let the computer do its job, and I was able to completely model the behavior of the floating arm trebuchet. As you can see in animations like this one, or this one for the projectile motion part. To effectively test my mathematical model, it was easier to focus on only one variable, which eventually led me to this research question, where I studied the relationship between the hook angle, which is how bent the hook where the string attaches is, as you can see in the diagram, and its relationship with the range, which is simply how far it shoots. And the sub-goal was then to find the optimal angle to throw as far as possible. As always in research and science, theories and models are nice, but the real deal are the experiments. This obviously applied to my project, which is why I had to build my own floating arm trebuchet to test the mathematical model in real life. I started with some prototypes, which I adapted into the blueprints you can see on screen, and you have the final product in front of you here. Then I measured the various parameters to input in the mathematical equations, and I finally performed an experiment in which I tested 9 different values of hook angle, which I tested 5 times each. Okay, now we can plot the prediction from the theoretical model, the dark blue line, alongside the experimental measurements, the little yellow dots. We can clearly see that they follow the same trend, but there is a shift of the peak. This can be attributed to the fact that the model currently ignores the effect of friction within the trebuchet. And based on this, the light blue line here is a qualitative modification of the theoretical model in an attempt to estimate the effect of friction, and we can see it fits a lot better. So, to conclude, my Lagrangian model is, as the results showed, qualitatively accurate at describing the relationship between the hook angle and the range of a floating arm trebuchet. But there is a quantitative shift due to unaccounted friction. As always in research, you end up with more questions than answers, so I still have lots of things I would like to dive into or improve in my current project, both on the theoretical and the experimental side. For example, on the theoretical side, as I mentioned, the model does not account for friction, which is something I could look into in the future, but it's quite complicated to do for reasons I won't get into. But I could also investigate any other variable that play a role in the model. On the experimental side, I would really love to build a huge trebuchet to throw very far, but I did not get the opportunity to do that, or simply a stronger one with better materials. What I really loved about this project, apart from the fun aspect of playing with a catapult, which you should not underestimate, is the same reason that makes me love physics so much. I find truly amazing how we're able to describe the behavior of the world around us with mathematical equations, and I hope I have convinced some of you of that. This concludes my presentation. Thank you for listening.
Thank you, Clément. That was very interesting, and we have some questions.
Yes.
When did you get this idea, and how do you choose to pursue it?
So originally, this came from a high school project I did, so probably similar to the Swedish equivalent of the high school project you mentioned earlier. So for me, I knew I wanted to do it in physics because it's a subject I'm really interested in. And why this specifically? It's really because I wanted to have fun with it, and a catapult is very fun. So this was the main reason.
Great. What are some advantages of using a Lagrangian model as opposed to classical Newtonian mechanics?
Newtonian mechanics uses forces to describe how a system behaves, and Lagrangian mechanics relies on energies, potential and kinetic energies, and this is a more general way of doing it that is applicable to more complex systems, because it would be very hard to model the forces acting in here, while the energies are just represented by a few objects like the counterweight, the projectile, and the arm. Mathematically, it's a lot easier to deal with to arrive at the equations of motion.
Cool. I saw earlier there was a curve you made that was for the friction. How did you create that exactly?
So this is just, let's say, preliminary estimate of how it would look like if I include it in the model. It was just trying to guess how, I mean, not guess, it is based on some work, but get an idea of how friction could influence the theoretical model and compare that to try to fit it to the results I got experimentally. Yeah, it was basically this, modifying the theoretical model so it fits the data and to get an idea of what friction could look like when I put it in the theoretical model with equations.
Awesome. Thank you very much.
Thank you.
Give it up for Clément! Are you bringing the catapult? Yeah.
Yes.
I thought I might get it, but no. Okay, we're up for the second presentation. This research investigates the process of leveraging large groups of rotating arrows to trace paths in 2 to 4 dimensional spaces. He is Swiss-German. Give it up for Jan Birmanns.
Hello everyone. I would like to spend the next few minutes talking about how you can use epicycles to create drawings in different sets of dimensions. But before I get started, let me quickly introduce myself. As you can tell by this huge picture of my face, my name is Jan Birmanns, and I've come here from a small town in Switzerland to represent the foundation of Swiss youth and science with the support of the Metrum Stiftung. When I'm not skipping classes to travel to Sweden, I'm a first-year student at the Swiss Federal Institute of Technology, where I'm studying math. This already brings me to the reason I'm here. Because of my interest in math, I've spent a lot of time throughout the past 2 years researching how one can draw with epicycles. But let me start at the beginning. What is an epicycle? For today's purposes, an epicycle is a rotating vector or arrow connected to another rotating vector, like right here. This might seem very simple at first, but we can actually use epicycles to create animations like this one. What has been done here is that I've taken hundreds, or I think even thousands of epicycles here, and I've picked their lengths and frequencies, rotations, such that the final one will trace this very specific path. And as you can imagine, the process of determining the lengths and frequencies of rotation of these arrows involves a lot of math. And I'll quickly touch on the most important aspects. At the center of this process is a formula called the discrete Fourier transform, which is extremely important for data analysis and so on, because it lets you take a periodic signal where you have one part that repeats over and over again and interpret that as a combination of many much simpler waves. Now, instead of applying this to experimental data, you can also apply it to a set of points that describe something like the shape of SIYSS. And the equation for that looks like this. And I know it's a little bit intimidating, but I'll guide you through this. So what we have here is a large sum that consists of many, many, many complex exponential functions. So they're called complex exponential functions, because they have this letter I in their exponent. So that little I you see over the E there, and this lets us apply Euler's formula, this thing right here, to turn all these individual summands into these values right here. And if you take a close look here, you might even notice that these little summons actually describe points moving along a circle. So what we get is this right here, a bunch of spinning arrows. And if we add these arrows together, we'll have something that connects the points at the beginning, but not very efficiently at all, right? And to understand why this is, let's take a look at just one arrow that is trying to connect 2 points. This arrow will, of course, create a circle, which it's okay, but really not a very efficient way of connecting 2 points. But if we take this arrow and cut it in half and have the 2 arrows spin in opposite directions, they'll create a line, which is way better. And if we take this idea and apply it to the epicycles we had before, and by this I mean have epicycles that now turn clockwise and not just anti-clockwise, we get a much cleaner result, right? There's still a few more tricks involved in drawing with epicycles in 2D, but there's also the process of moving to 4D and using them to draw there. This is what that can look like, and I'll quickly explain why exactly this is 4D. So any point in 4-dimensional space is uniquely defined by a set of 4 different values like A, B, C, and D, like with 3D, X, Y, and Z. Now to visualize these 4 values, there's millions of possibilities. What I did here is that I took the first 3 values, A, B, and C, and took the corresponding point in 3D space, and then took the 4th value and translated that to a color, and then gave that point the corresponding color. So now we just describe these 40 points with colored points in 3D space. Describing these points mathematically can be a somewhat different story. I used quaternions to do this, which are just, for today's purposes, a very compact and elegant way of describing these points. And of course, since we're now talking about 4D, the method I showed you before has to be modified slightly. And what we have now is the discrete quaternion for a transform, again, bunch of numbers. But the main difference here is that what we get out at the end are a bunch of arrows that are turning on planes in 4-dimensional space. And if we take a look at one of these arrows and maybe forget about the meaning of the color for now, you might be able to tell that this arrow has a length of slightly over 2 units at the top and slightly under 2 units on the sides. And this is because this arrow isn't really an arrow anymore. It's actually, or I like to refer to them as elliptical epicycles because that's the very shape you see right here. And unfortunately, there's no way of actually completely eliminating this effect, this elongation of the circle. However, when we're describing the 3D path at the beginning, it works best if we pick the unused dimension to be as small as possible, like 0, and then we'll get something that looks more similar to a circle. If any of you would like to draw with epicycles yourselves, as a part of my project, I created these 2 websites right here, dft.birmanns.org and dqft.birmanns.org. The first one will let you experiment on a 2-dimensional plane and draw with epicycles that way. And the second one, dqft.birmanns.org, lets you plot points in 3D space, which will then be traced by these elliptical epicycles that I mentioned earlier. I hope that I was able to show you that math can lead to some very fascinating results that arise from these great equations, even though you wouldn't expect it at first. And I, of course, appreciate all of you for letting me share this topic with you, and SIYSS, the Foundation of Swiss Youth in Science, and the Metrum Stiftung for giving me this opportunity in the first place. Thank you.
Fascinating. Time for some Q&A. One question that popped up was, what sparked your interest in epicycles, or I guess Fourier transform in general?
Sorry, can you repeat that again?
What sparked your interest in epicycles?
So the 2D application is something that is somewhat well known already, and I saw that online. And at the same time, I read a paper about quaternions, and I figured that one could somehow combine the two, which is what I spent most of my research on. And I also improved the method for 2D and kind of saw where things went and just found it really fascinating.
Do you have any advice for aspiring researchers interested in math?
Don't be too intimidated by these very complex equations and all. It's just, like, at first, yes, they are very complicated, but if you just go step by step and learn bit by bit, you'll get there eventually. It's not that challenging.
Great. Thank you so much.
Thank you.
Give it up for Jan! This next project focuses on creating a vaccine against dengue virus. He is from Mexico. Give it up for Raul, Acosta, and Murillo.
Thank you. Well, first of all, hi, my name is Raul Acosta Murillo. I come from the northern part of Mexico, and I came here to present you my project called In Silico Design and Production of a Multi-Epitope Peptide Vaccine Against Dengue Virus. So first, let us talk more about the dengue virus. So we know a lot of people are at risk. Only the continent of the Americas, 500 million people are at high risk of developing dengue. In the whole world, this sums up about 4 billion people, so half world population. Then the dengue virus, it's a pretty complex problem, and it's aligned with the Sustainable Development Goal number 3 and the World Mosquito Program. Also, we know there's a lack of effective prevention and protection against it. Until some weeks ago, the only available vaccine required you to be prior confirmed case of dengue in order to be applied the vaccine as well. And we also know there's a high probability of severe disease in case you got reinfection with another serotype of the virus. So which were the steps we took in order to design this vaccine? In order to sum it up, there were about approximately 40 different databases and tools we used to design it. But first, we obtained the sequence of the virus. So we use a database called UniProt. In this database, different scientists report the sequence of this virus in the whole world. So we took approximately 1,000 sequences. Then we evaluate the efficacy and the antigenicity of these epitopes that are regions in which the antibodies bind. Also, we build this multi-epitope vaccine, and then we evaluate the physicochemical safety and efficacy of this. We also simulate the structure, and we simulate the immune response. So what did we find? Well, actually, we found 25 regions of the membrane virus of the dengue, which were highly immunogenic, so they have a high probability of creating an immune response. And we also searched the ones that were safety. So they didn't have any similarities to human proteins, and they didn't show any similarity to known allergens or toxins. Also, in this case, we also add 2 adjuvants. So we have coadjuvants or adjuvants that are regions that help us increase the immune response. In this case, even if the protein we designed wasn't a virus as itself, it still stimulates the body as if it were one. So then we actually designed a gene, a synthetic gene that was included all the parts of specifics to E. coli in order to produce it in a live environment. So we create a synthetic gene with a strong constitutive promoter and help us produce high quantities of this vaccine. In this case, as you can see, we produce and we used a high copy number plasmid in order to produce it constitutively. So we can see the structure of the protein itself. In this case, if the protein is produced and purified in this bacteria, we could inject it in animals for testing or in testing its neutralizing antibody production. In this case, if it were the case in which this bacteria functions correctly, we could pass to another level of these lab tests, and well, in the future we could eventually patent or help sell this vaccine. In this case, this is the simulation of the immunogenic response. We can see how the antibody production, in this case the different types of immunoglobulins, increase after the first vaccination. Also, we have the populations of the B cell memory cells, in this case that are the ones responsible to identify in a further environment and activate the latter production of antibodies. And Also, it is not shown here, but we also follow the populations of CTL cells that are cytotoxic cells and HTL cells that are the helper or cooperative cells. In this case, these three, CTL, LBL, and HTL, could regularize and arrange or control the whole immune response. So we did the simulations in a site called CIMSIM, or Computer Immunogenic Simulations, and in theory or in paper it should work well, and in future steps we could continue with the production and evaluation of this protein. So, well, this is some of the literature we use. There are approximately, the paper expanded review includes like 35 pages, so it's a bit of a sum up of what all the job we did, but I think we had pretty good results with the simulations. Thank you.
Thank you, Raul.
In silico vaccine design, fascinating. So one question I got was, why dengue virus specifically?
Well, we actually need to study in the genetics of the virus, there are certain regions that are shared. So we have these 4 serotypes of the virus. And you remember in COVID, we had like Omicron, Delta, and all of those versions. So we did study the conserved regions of the dengue virus and use them as a specific way of protecting against this.
Interesting. I also got a question about the databases you use for protein sequences. Are they public?
Yeah, actually we use mainly public databases in which scientists all around the world publish their results. So we wanted also to make public our results, and I made a Python script for automating this process, and it's also public in GitHub. You can access it, and if you give this program your amino acid sequence, it can generate you a design of a vaccine against it.
Wow, that's really cool. What were some major challenges you encountered during this research?
Well, one of the main challenges, at least in the in silico part, is the accessing specific tools, like finding first the tools you will need, then identifying the parameters, because all these applications have default parameters and optimized parameters. So optimizing the parameters for your own project, and well, at the end, the simulation, the computational power, and all those stuff.
Thank you so much, Raul.
Thank you.
Yeah, give it up for Raul. This next research focuses on improving the long-term performance of neural probes. Give it up for Kanghyeon Kim from the Republic of Korea.
Thank you. Okay. I think you cannot say the razor well, but okay, I'll go. Hi everyone, my name is Kanghyeon Kim and I'm from South Korea. So today I will talk about novel designs of neural interface devices and the computational simulation with the final analysis. Yeah, I know it's quite long and difficult, but yeah, I will explain this well. Sorry. Okay, first of all, I will introduce my research history and the topic of research. So this is my first research, so it is not STEM. So my dream is to make a world where everyone can enjoy their lives. So I studied the inequality at the geopolitics office and conclude that the most important problem for this world is the health equity. So I focus on the health equity. So I work in the medical device startup, and I try to find the ultimate challenge in the medicine. So there are many innovative biotechnologies, including some artificial organs or gene editing. And I found that the brain, which is the core that determines our identity, cannot be replaced or edited. So therefore, my research is drawn to the brain. brain. So I studied the brain and the tool for this, neural interface. So a neural interface is a tool for recording or imaging or stimulating the brain. So we can understand or interact with the brain with this tool. The major part of the neural interface is the neural implantable device, and neural implantation causes some immune reaction. So as a result, the implantation causes the glia skull, which is the green part in this figure. So it acts as a barrier between the cell and the probe. So actually the probe needs to get the signal from the cell and also give some electrical signal to the cell, but the barrier blocks the function. So it causes some loss of functionality. So this is the main challenge I want to address. And then when I see this situation, there is some mechanical background. There are two main causes of the mechanical. The first one is that mechanical mismatch between the neural probe and the brain tissue. Actually, the conventional neural probe is made of the silicon or metal, so which have the elastic modulus of 10 to 100 gigapascal. But the brain tissue has the elastic modulus of the 1 to 10 kilopascal. So it's kind of that, you can imagine that the neural probe is similar to the fork, and the brain tissue is soft like the pudding. And then the second one is the brain micromotion. So there are two causes for the brain micromotion. The one is the vascular pulsation, and the second one is respiration. And then, so I can say that the implanted situation is same as when the fork is inserted to the pudding and the dish is shaking down. So it causes the immune reaction, and immune reaction causes the loss of functionality. So to predict how these devices affect the brain mechanically, we use the method called finite element analysis, FEA. So with FEA, we calculate the macro situation by dividing them into small problems. And previous studies had some critical issues. The first one is that there is no verification of the model with the histological analysis. And also, they only used limited designs, such as some conventional shank design. And the third one is that they using uncertain logic situation, so they temporarily fix like 1 micronewton for the brain micromotion. Therefore, here is the from my study. So the firstly, I fabricated the soft fibers with the TDP. So actually, I bring some sample of the microfiber, but I'm not certain you can see. And this is the neural fiber. So I implant this fiber into the brain, and this is the preform. So you can see the design, and maybe it is macro scale, so I can make some channels or electrodes into them. And when I get the heat and cool down, then I can make this micro scale fibers. And I will implant this the mice brain. Also, I will make the simulation about this situation. So IHC, so I mentioned about the FEA in the previous slide, and IHC is some technique used to detect specific proteins in tissue using the antibodies which bind to them. So it's just investigating some immune response in the brain tissue. So results. The results show that, okay, I think the results also show that, sorry, the results show that the soft material causes decreased immune response. So you can check the result of the computer simulation. So especially the hydrogel neural implant, so the much more safe situation than other implants. So we can check the potential of the hydrogel, and to addition, I can also check that the they showed a similar trend between the computational simulation and the actual immune response. So it demonstrates the effectiveness of our model into this situation. And as a result, also they said they showed the lower coefficient friction and higher aspect ratio and sharp design caused a higher immune response. So this is the maybe potential strategy for the new designs for the neural interface. So if you want more information, you can check this from the below paper. Thank you for listening.
Wow, really interesting. So, I'm sure many people were inspired by what you mentioned at the start, your reason for pursuing this research. I wonder, do you have any advice for young people who want to make a change in the world?
Sorry, can you repeat the question again?
Do you have any advice for young people who want to make a change in the world?
Advice. Okay, when you research or try to change the world, I think the really important thing is not the talent, but the core is that the long-term interest. So when you have some interest in some specific questions, then focus on that, and then keep it on a long time. Then you will succeed. That's my advice.
Thank you. Do you think these brain-computer interfaces can pave the way for research in neuroscience and understanding the brain better?
Yeah, actually it is actively used in the neuroscience for brain mapping or some treatment for the Parkinson's or Alzheimer's or like that. So yeah, it is actively using that. Yeah.
Awesome. What part of your research was the most fun?
Actually, I'm focusing on the clinical aspects, so like treatment for the neurological disorders. So when we get some perfect treatment for the neurological disorders, then it means we can maintain our brain activity forever, and it will change our lives. Yeah, I think.
What part of the process was most challenging?
Sorry?
What part of the process was most challenging?
Actually, I put together the team with the other laboratories. So I'm supposed to get the brain micromotion data from the other lab, but COVID-19 shut down the lab, so I cannot get the data. But I can make a simulation based on the other data. So I want to say that I cannot make the future, but I can adapt to the future. Yeah.
Awesome. Thank you so much.
A big round of applause for Kanghyeon Kim.
Awesome.
Okay, so this was the fourth of 4 presentation in this session. So we have now listened to all of them. Thank you so much for your engagement, for your big round of applause, your SIYSS 2023 applause. And I want to really encourage you to take that opportunity that Elin shared in the beginning, that if you are doing some kind of science diploma in your high school, then participate in the exhibition at Unga Forskare. You can log on to ungaforskare.se to know more about that. So from this stage, we will take a break and we will begin again with the roundtable discussion, the panel discussion, Minds of the Future, starting at 2 o'clock. And there will be a full house. Every seat will be taken in here by then. So you can stay if you want to, but you can also go out and be back. But be back 15 minutes before 1:45. And we will have a really, really inspiring conversation together with an honorary guest. So see you at Minds of the Future in a short while. Thank you.